Okay, let's pick up where we left off. Piggies. Okay, so if you recall, last thing we talked about was code, right? So obviously it decides whether or not it feels like saving the notes or not, but they're all on the recording. So I'll have to just consult that one. Yes, I know I still need to get those posted, so those are coming shortly. So that was, we kind of learned about our genetic code last time. And so the other big thing that is important about the genetic code that we realized was that it's, for the most part, it's a universal code. But basically means is that we all essentially have the same plan, from bacteria to us, to pigs, to mice. There's, as it turns out, really only one plan for life. And we just have slight variations of that basic plan. You may recall when I played Mozart's variations on Twinkle Twinkle Little Star in the lab, right? So there's like one melody, right? Twinkle Twinkle Little Star, but you have 12 versions of that, right? So 12 variations of it. So you would argue that essentially the melody is universal throughout that entire variation. Same thing's true here. It's not that they're identical, it's that they're universal. And that actually turns out to be a good thing, right? So in terms of sort of the evolutionary side of things, this is kind of like where we kind of get this idea that it's universal because that reflects common ancestry, very similar to your common ancestry in your families, right? So basically you and your siblings share commonalities. You have common traits because you have common ancestry in the same family, right? Your parents are the same. Well, for the most part, in case you have like half brothers or whatever. But still, you have a parent that's the same, right? There's a sense of common ancestry, and so that's kind of what we found out here. But what really makes this important is that it allowed us to be able to do genetic engineering. And this was actually kind of a very, very happy surprise because we were bracing ourselves for, okay, great, how many different species out there? And there's like a million trillion different ways of doing the same thing. So we were thinking we had our work cut out for us. And as it turned out, it was all largely pretty much the same. Now, what does this mean in terms of genetic engineering? Well, what it means for us is that we can actually do a lot of molecular advancements. This is an example right here. We can do what's called transgenics. And basically what transgenics is taking a gene from one organism and putting it into another. Like, for instance, our little piggies here. Right. So this little guy right here, that's your transgenic pig. This is your normal pig. And as it turns out, actually transgenics has really, really helped us advance our knowledge. And really, a lot of people don't realize as they're picketing GMO and stuff like that and wanting to do away with all of that, they don't realize that if we do away with all of that, we'll pretty much take all of our scientific advancements and pretty much flush them down the toilet. So we'll basically go back into a technological and medical black hole. Okay. Hello, Dark Ages, good old friend, right? So it's where we unlearned everything we learned. All of our medical advancements are essentially benefits of the same idea. So what does that mean? So what that basically means, then, anytime we learn something about genetics in some organism, fruit fly or 4 o'clock plants, as the case may be, because of the universality of the code, it means that those lessons learned in that organism are immediately transferable to humans, which is the way we study ourselves, right? We don't study ourselves by experimenting on ourselves. Last time I checked, I was still against the Geneva Convention. We generally don't like to see people experimenting on other people. That's usually a bad thing. It's usually frowned upon. I think the last major political figure that actually tried to do that, at least in Europe, was Mengele. And well, we all know what his nickname was, perhaps, or do we? His nickname was the Angel of Death. So that's actually what his nickname was. He's a horrendous creature, a malevolent, dark piece of work that hopefully humankind will never duplicate again. But will, unfortunately, because that's just who we are. So this is basically what it does, right? It basically allows us to be able to sort of use things like mice, right? and be able to work with them genetically and develop therapeutics in mice so that we can actually utilize those and move those into humans. All right, this is actually how we develop medicines. This is how we develop drugs. Not because we're picking on mice and we hate mice, right? I'm looking at you, PETA, right? Get your signs off my lawn, right? Is because if we didn't have the chance to do that, then we wouldn't have any of the medical breakthroughs that we have. And unfortunately, that's the other side of that story that oftentimes PETA ignores, right? That we're not trying to pick on mice. Matter of fact, I actually worked in a mouse lab, and it is exceptionally regulated, highly regulated by the government, more so than humans. We actually treat in mouse labs, the grad students are actually treated worse than the mice. I mean, the mice have it good, I mean, with the exception of having to sacrifice them from time to time. But the protocols for sacrificing them are very rigid. Like, you have to do it a certain way so that you're not inflicting harm on the mouse, like you're not tormenting or torturing the mouse or things like that. So it is exceptionally regulated. We probably do a lot worse in our prisons, honestly. So I just always like to sort of get a little dose of perspective in there, right? So the next time PETA pickets outside our labs, it's like, why don't you go to the prison and actually talk about the treatments in there, right? Because they're doing a lot worse than what we're doing with mice. But this is basically a transgenic pig, and it kind of just to kind of explain to you what this means, right? How we actually use this, and it was a huge breakthrough. Is we want to go back to where we talked about oncogenes. Remember, we talked about oncogenes and tumor suppressor genes, and we talked about these genes that basically were in charge of cell division. Well, in this particular case, when you take a look at our little pig friend here, it looks like it's some sort of a skin gene, right? It's making some sort of a protein. If I had to guess, I'm probably thinking keratin, because keratin, that's the major structural protein in your skin, your hair, and your nails. This looks about where I would expect to see keratin distribution in a pig, because the snouts are actually really kind of tough and keratinized. So are the hooves, right? Because those are like little pig nails. And if you take a close look at the hair's kind of got a little blondish look to it. So in this case, you have some sort of a skin protein. And of course, it's under the control of a promoter, right? So when we take a look at the control of this promoter, and this is kind of one of the big revelations that we noticed with these gene expression studies, was that when we take a look at a protein and we realize that a protein is actually, it's basically made in, one particular area or at one particular time in development, what we noticed is what's driving that pattern is not the gene itself. We used to think that. What's actually driving that pattern is the actual promoter. And as it turns out, it's irrelevant what's behind the promoter, because you could put anything back there and the promoter will turn on and turn off at the time it turns on and turns off. That turns out to be quite helpful, because what we did was we created our little yellow pig friend here, and we swapped out the promoter, and we kept the promoter, but we put behind it, instead of a skin gene, we put a reporter. And a reporter gene, like GFP, which is green fluorescent protein, is essentially a protein that when it's expressed, we'll be able to visualize. We can actually see it. Green fluorescent protein, by the way, is a protein that we get from jellyfish. And it's the protein that will basically make jellyfish luminesce. So if you shine a black light on it, UV radiation, it'll go through that whole absorption of the radiation, excite the electrons fall down, and it basically will fluoresce out, in this case, green pigment, right? It's kind of what creates that sort of greenish glow that these jellyfish have. So what we did is we took GFP, and instead of the skin gene, we basically swapped it out and put it behind this skin gene promoter. And what that means is, now that you've got a visible protein there, you put it back into the pig, you let it kind of distribute throughout the pig, and that promoter just turns on like normal. It doesn't know anything's different. It just turns on wherever it turns on, whenever it turns on. And what you get is a visible distribution of your reporter protein in the exact position at the exact time that you would normally have your skin protein produced. So it's really, really helpful, because it tells us visually where this protein is, when it's doing its work, and it gives us a lot of information about this protein. Yeah, Kate.
Would the pig with the GFP be more vulnerable to
No, because everything is the same. All you've done is literally swapped out a different protein. And so everything else would be normal in the pig. So in this case, you can see you shine a little bit of a black light on it, and you've got your reporter gene glowing wherever that protein normally would have occurred. And so now I know that this particular protein is showing a similar distribution that I would expect from keratin. So this could potentially be the keratin gene. Right. And you notice this guy here, he's kind of a little it's not quite pink, it's kind of a little purplish pink. That's because of the UV that's shining on this little guy. That's why he's kind of more of a little purplish pink to him. He's not quite that pink. Is that a question? I saw a hand going up. Okay. The promoter is part of the regulatory sequence that's basically upstream of the actual gene itself. So inside the box, we'll talk about this later on, is basically all the coding sequence that will essentially become amino acid of the protein itself. Everything above that, which is what we refer to as upstream, is basically all regulatory sequence. So it's just all how you regulate how this particular protein is made. Okay, we'll talk much more about that. But let's dig in. And so this is really actually an innovation that was just like crazy, because now what we can do is we can watch and study proteins and what they do and where they are, and be able to understand a lot of information about them. Okay, and this has really been helpful. And it's been so helpful, as a matter of fact, that actually the folks that originally isolated GFP and utilized it as a reporter actually won the Nobel Prize a few years ago. That's how significant of help that this has been. Not only that, but one of my favorite things, actually, you can actually Google this, transgenics, in terms of transgenics, is actually older, predates GFP actually. And it was done in the 90s, or when I was still in college, ever they did it. It was like, whoa, my gosh, it was crazy. About the time when they had Dolly, the sheep, the clone sheep, is around the same time. But it's the glow-in-the-dark tobacco plant. Love this one because they used a different reporter called luciferase, which is an enzyme. And so you kind of give it a substrate and it'll bioluminesce. But luciferase is the gene or the enzyme that's responsible for making fireflies glow. So like their little butts are glowing because of luciferase. So what they did was they took some sort of a plant promoter from some sort of a gene. I don't know. It was expressed throughout the entire plant. Hooked it up to luciferase, put it back in the plant, let the plant grow up with it, treat it like its normal gene, and then they fed it the luciferase substrate, and they turned out the lights. And what you see is this glow-in-the-dark tobacco plant. It's awesome. Google it. You can actually see it. Glow-in-the-dark tobacco plant. There's images. They did a lot of studies with this. It was one of my favorites because that was like one of the first transgenics that you're like, whoa, that's what you do with transgenics. That was really kind of cool. Now, what's happening with transcription, right? So let's go ahead and take a look at transcription. We're going to take a look at prokaryotes first, and then we're going to be moving into eukaryotes. So the first thing to understand is that in prokaryotes, you only have one RNA polymerase in prokaryotes. We'll see how that's different in eukaryotes because we have 3 of them. But ultimately, when you take a look at a prokaryote, you have the same kind of a 3-step system, right? So steps of initiation, you have steps of elongation, and of course you have steps of termination. And so you have all of those in play. A couple of things that you absolutely need in order to get this off the ground is you need a promoter, right? You need a start site. You got to know where to start, and then you need a termination site. And those are 3 necessary things in your transcription. Notice 2 of these 3 are associated with initiation. Right, so those are all things that you need in order to get yourself off the ground. Of course, you need a place to stop, and that's what we noticed with prokaryotes. Now we take a little closer look at the promoter itself. First thing is the promoter. One of the big jobs the promoter has to do is it has to do recognition, right? So you actually have to recognize and be a binding spot for the RNA polymerase, right? So think about this. Remember, DNA is DNA, right? It's all a series of ACGs and Ts. So the question is, how do you know which part of the ACGT is going to be coding sequence and which part is not? Which part is the promoter? Well, the way that you actually have that is with actual specific sequence. So specific sequence that is invariable is oftentimes referred to as defined as consensus sequence. Bacteria have a lot of this. It's like little signals, right? Like little sequence signals that tell you this is me, right? This is what this is. Very similar, remember when we talked about restriction enzymes in lab, and we talked about how they have specific sequences that they recognize? That's a consensus sequence. So it's like use a particular sequence of DNA bases that sort of trips and signals a particular thing, right? Good. Oh yeah, yeah, it does happen from time to time, and that would be a particular type of a mutation that we would probably uncover. But yeah, you have mutations in the regulatory region that affects a gene's expression, as well as in the actual sequence of the coding sequence itself. So typically when you see those mutations, you'll notice that it's not necessarily like a foul up of the actual protein itself, where you're missing an amino acid, or you've got sort of like a misassignment of an amino acid. But in this case, it's actually more of like, it's like a foul up in sort of the regulation of the protein itself, right? So like normally we should have this much protein, but we don't have it at all. That suggests that perhaps there might be a regulatory region. So you can't actually put it all together to make it, because you've fouled up that regulatory region. So now generally we also have what's called the start site. And these are also in the regulatory regions, and they're not transcribed, right? So this transcription piece of it is associated with actually what is, so in bacteria, you have your transcript, and a portion of that is going to actually be coded for and actually be assigned to amino acids. And there's a portion of it that's more regulatory. We'll talk a little bit about that a little bit later on when we get a little further on in the chapter. But one of the things we're going to do before that is we want to kind of take a look at this promoter region and sort of crack this open a bit more. First of all, before we do that, we have to introduce ourselves to the RNA polymerase of bacteria, which has multiple subunits in it. And because it's a big complex and it's only doing one thing, RNA polymerase stuff, right? That's all it's doing. We oftentimes refer to that as a holoenzyme. So anytime you hear the term holoenzyme, that usually means it's a big thing. It's like, this is just a big complex with a lot of subunits in it, but it's really only doing one reaction. In this case, it's making RNA. So you'll notice how you have your two alphas here, you have your two betas here. That kind of forms a core polymerase. But the real biggie that you're going to see is this guy right here. That's your sigma subunit. It looks like an index finger and a thumb, like little pincher fingers, right? That's basically what you've got here. And those little pincher fingers actually become important in the mechanism of how RNA polymerase works. So let's take a look at this first of all. So on the right-hand side over here, you're going to notice we're going to first of all define our start site. Now, typically speaking, you can always define it in molecular biology. We define our start site as a +1. What that means is it's the first nucleotide in the transcript. That's what +1 means. The second nucleotide in the transcript is +2. The third nucleotide in the transcript is +3, and so forth. Okay. Now then, the nucleotide that's just on the other side. So if you're going in this direction, we usually refer to as you're moving downstream, because that's usually the direction of transcription. If you're moving this direction, we usually refer to this as upstream. So sometimes I think you probably heard me mention that a few times, upstream and downstream. That's what I'm talking about. You're moving to the left, it's upstream. You're moving to the right, it's downstream. So immediately upstream, the very first nucleotide that is not part of the transcript is considered -1. The second one further on is -2, -3, and so forth. So we have a numbering system to kind of help us positionally orient ourselves with respect to the beginning of the transcript. Now, why is that important? The reason why that's important is because when you take a look at your sequence that's -10, that is to say 10 nucleotides upstream of your start site, you have what's called the Tata box. And this is consensus sequence. All promoters have it. It's a TA-rich region. And this is where your index finger of your sigma subunit will bind. It grabs onto the Tata box, this TA-rich region. Now, it's consensus sequence in the sense that it's TA rich. It's not like, okay, it's got to be TAAT. It's a TA rich region. So it could be TAATAT, right? Or variations thereof, but it's a TA rich region. That's the consensus component of it. And so all promoters have this, not just in prokaryotes, but also in eukaryotes, in all genes, and all taxa. As a matter of fact, when we are looking for a promoter in C. elegans in the research lab, we are looking for a Tata box in all cases, because that is so tight and so well conserved that we see it as a marker, sort of as a red flag saying, hello, I'm a promoter, when you see that Tata box. So your index finger of your sigma subunit is going to grab onto that -10 region right there. Now, if you kind of move a little further upstream at your -35 section, you're going to have another little consensus sequence. It's not quite as strong, right? So it's kind of like a TGACA kind of a complex, a varying versions of this. And then this is where your thumb portion of your sigma subunit grabs onto. So with your index finger, you grab onto the Tata box. With your thumb, you grab onto the -30. And so you got a hold of your promoter. So now your polymerase is officially grabbed onto it. But it doesn't stop there, right? Because we all know from replication that one of the things that you have to do once you grab onto this is you have to unwind your DNA and pop it open, right? We had helicase to do that for us last time. There is no such thing this time. So how do you get this unwound? Well, as it turns out, once you bind onto it, this is this piece right here, then it's going to happen is you're going to unwind the DNA. How do you do that? With your sigma subunit. So literally what you're doing is you're kind of grabbing onto your DNA and you're kind of like working it with your index finger and your thumb, and you're unwinding it with your thumb. If anybody's has birds, parakeets, we've got a lot of them. We got 10 of them actually. So we got plenty of them. But if you ever watch closely, the way a parakeet, a seed eater, eats the seeds, they grab it with their little beak, which is like little pinchers, and then they're kind of working it with their like jaw like that. They're kind of cracking open the husk, and they're kind of getting out that little kernel. You can see this also if you have bird feeders with birds who like to work on sunflower seeds. You'll see that a lot too. They'll pick up the sunflower seed, right? Of course, the good stuff is in this inside, right? You'll kind of see them working it, and they're kind of like just sort of like chiseling that away. They're using like their upper and lower beak, sort of like your pincher fingers, and they're kind of cracking that open a little bit, and they're getting that seed out of there. And that's exactly what you're doing here with your sigma subunit. You're sort of unwinding that little piece of DNA to create that little open region, which we refer to as a transcription bubble. So guess what? All of this, initiation. You haven't even done anything yet. You've just been preparing for it. You just basically been finding your promoter, binding to your promoter, unwinding your DNA, getting your transcription bubble set up. Now you're ready to roll. But just when it starts getting good and you get your transcription bubble open, guess what? There goes your sigma. So you lose your sigma and your core polymerase is now in charge. But notice, you maintain your transcription bubble inside your core polymerase. So really, your sigma subunit was purely an initiation factor. It was there to bind you to the promoter, to unwind your DNA, to get your transcription bubble set inside you as a polymerase. And once you look good, got everything you need, polymerase? Okay, good. I'm out of here. Right? I got you right where I want you. Now I'm gone. Okay. And then at that point, it becomes elongation. So what does elongation look like? Well, it kind of looks a lot like replication, only in this case, you're making RNA, right? So you're still going 5' to 3', only in this case, you're using ribonucleotides instead of deoxyribonucleotides. You're also doing everything inside your transcription bubble, which looks a lot like a replication bubble in eukaryotes, only it's inside your RNA polymerase. So what's in this? Well, obviously, you've got your RNA polymerase. You've got your DNA template. Remember, you're only reading one template, one side of the DNA at a time. You're not reading both, just one. And then, of course, you got your RNA transcript. Now, the important thing to understand is that your transcription bubble basically is rewound before it leaves the RNA polymerase. So what happens is your RNA polymerase will basically, as it's sort of moving down, it will basically take in the DNA, unwind it into your transcription bubble, make your copy of RNA, and as soon as you're done, guess what? You don't need it anymore. So as soon as you're done, it'll twist it back up on this end, and then you see the DNA come out the RNA polymerase. So if you were looking at it from the outside, looking in, all you would see is twisted DNA duplex coming in, twisted DNA duplex coming out. You would never actually see the replication bubble where it's unwound. Why is that important? Because it protects it from nucleases that want desperately to devour and destroy single-stranded DNA template. So that's kind of what it looks like. So here you can see you've got your replication bubble here. So it's unwinding it as it comes in. And then once it unwinds it, it basically makes a copy of it and spitting out your mRNA template, and then it rewinds it once it's done. So basically you never actually see it. It's not undone for very long, right? So it's considerably less risky in terms of exposure than, say, DNA replication, where you've got these big old replication bubbles all wide open, splayed open, and you need single-stranded binding protein to protect them, right? Which is oftentimes a big question I get a lot of times. This one, a lot of times the students will be like, wait. How come you don't have single-stranded binding protein attached over here? Right? I mean, aren't these guys? Well, it's because they never see the light of day. These are carefully and closely and protected, sequestered deep inside the RNA polymerase. It never sees the outside world until you twist it back up, and then it's fine. Okay. So termination for prokaryotes, where we're still in prokaryotes, basically has a stop signal that basically tells it when to stop. And so at that particular point, once you hit that stop signal, you're going to stop making phosphodiester bonds. You're going to have a little bit of a RNA hybrid, right? Because your RNA will kind of be stuck there for a second and be like, okay, wait a minute. So are we still going? No, we're sort of stopped there. I'm still kind of tethered to my template. And then eventually what's going to happen is you're going to release everything, and you're going to rewind up your DNA. And then your RNA will form a hairpin. What does a hairpin do? It protects the RNA from digestion. So basically what a hairpin is when you have two regions of complementary sequence and they basically zipper up, they kind of fold back on itself. And you create this like hairpin structure. Okay. And what that does, it protects the RNA from being degraded. So that's quite helpful. So here's what it looks like. So here you can see you've got this little stem loop hairpin thing. And the signal here is poly U, so you get a stretch of U nucleotides that kind of destabilizes the system and sort of causes it to fall apart. And once it falls apart, it releases the DNA, and then it starts over again. It goes and does it all over again, right? So there's that one. Let's see if there's anybody who is joined us. Nope. Sorry, my OCD come in. Okay. So then transcription translation in prokaryotes is something that's coupled. It's coupled spatially. So think about it. In a prokaryote, it's wide open in the middle, isn't it? There's nothing in there. It's just empty cytoplasm, and that's it, right? Think about this for a second. Let's compare it to a eukaryote. Where do you think transcription is happening in a eukaryote? In the nucleus, right? Because that's where the DNA is. So that's where you have to make a copy of it. But where does translation happen? Yeah, right, because the ribosomes are needed, and where are they? They're outside the nucleus, right? So spatially, you're separated in eukaryotes. So can you do transcription and translation at the same time? Because they're spatially separated, right? But bacteria can, because they don't have a nucleus. So when they're doing transcription, literally, there's a ribosome sitting right there. And it's like, oh, wait a minute, you're kind of starting to thread out this little transcript, and your ribosome's like, can I go ahead and get started on that? I mean, I know you're not done yet. You keep doing, but can I get started on that? So the answer is yes. And so it basically gloms on there and just starts working on that before transcription even stops. So transcription and translation are happening essentially simultaneously in bacteria. Why? Because it's all happening in the same space. So this is helpful for bacteria. Why? Because they have an organization in their genome where they organize their genes in expressed clusters called operons. So this is basically an organization where genes that are functionally related are expressed at the same time. So this is helpful, right? Because imagine, let's say you have 3 genes that are involved in the same biochemical pathway. Genes A, B, and C. And so they were all codependent on each other, right? So the bacteria basically is going to have a promoter that drives all 3 of these. So when you make a transcript, it's going to basically make a transcript with all 3 of them on there, A, B, and C. This is what's referred to as a polycistronic mRNA. So basically, when you make one transcript, you get all 3 genes, which is nice, right? Because they're all related and they're all basically at work in the same process. It's just A has step A, and then B has step B, and then C has step C. And so you need all 3 of them in order to work. So it makes a lot of sense to make them all at the same time, right? And that's kind of what bacteria do. So when they basically are doing coupled transcription then and translation, then they can basically power out these related, function related genes all at the same time. Brutally efficient. This one, polycistronic. That's a C. So that's how prokaryotes do it, though. And that's a common organization that we see. And there's a lot of advantages to that, right? It's very, very efficient. And it's good so long as you don't need greater nuance in how you regulate these 3, right? If you're always going to need all 3 of them at the same time, this is really nice and efficient, right? One step, everything's good. That's not how it works in eukaryotes, though. In eukaryotes, basically, what you have are 3 different genes under 3 different promoters. So as a result, you can have a lot of A, but a little bit of B, or a moderate amount of C. So you can attenuate each one of them, and that'll give you different effects. That's helpful if what you need is not just one blanket response. You need a little bit of attenuation or modulation in your response. I like to think of it in a lot of ways, the difference between operonic organization in prokaryotes versus non-operonic in eukaryotes as sort of like theater lighting. Anybody who's a fan of theater and lots of different performing arts, especially if you pay close attention to set design, there's a lot of awesome things that you can do with theatrical lighting. I mean, actually, in some performing arts colleges, it's like theatrical lighting is like a degree in its own right. Why? Because you're not just flipping on lights or flipping them off, right? Every single light has its own control. And a masterful lighting person can really create a lot of magic with just the way they're attenuating the light. They can create so many optical illusions with just lighting that it just kind of blows you away about how good lighting can be in theaters. A lot of people don't realize that. But a lot of the cool stuff they see in theaters and set design is not so much the set as much as it is the lighting that kind of creates all these weird and wonky effects. But that's kind of what eukaryotes are like. Eukaryotes basically have much more attenuation and separate controls so that they can respond to different situations. A good example is this room as well. Our light switch, we've only got one in here, don't we? That means the lights are all on or all off. But what if we had a lot of glare here, right? Well, it's a little dark in the back for you guys, right? But what if we wanted a lot more light back there, but less light up here? Guess what? We're screwed, aren't we? Unless we break that up and put our banks of lights on separate switches, so we can differentially control what we have. So maybe I can have those lights, you know, on bright so you guys can see. But up here, I can have these guys dimmed down so that there's no glare against the board. That would require different controllers for different batteries of lights. That's exactly what eukaryotes have. So lighting wise, you can create a much more nuanced atmosphere that basically serves greater functions. And you can do more with it than just lights on, lights off, right? So that's kind of what bacteria do. It's like it's an all or nothing sort of a thing. But eukaryotes can have a lot of nuances in them. So this is an actual electron micrograph of coupled transcription translation. So here you can see you got your RNA polymerase, and they're talking about that little guy right there. And then it's basically making a transcript. It's not even done making the transcript, but these little black blobs on here that you see, those are ribosomes. They're already glomming on to the transcript itself. And away they go. So you can see the longer your transcript gets, the more ribosomes glom on there. And then eventually these ribosomes start to make their polypeptides. And you can see these proteins basically coming out as soon as the transcript is coming out. So this allows you, so in this case, you got a really, really efficient system to produce a lot of whatever this protein is in a very short period of time, right? You don't have to wait for this to go find a ribosome, and then you'll make one and then falls apart, and you go make another ribosome, and you go make another one. So it's not like a sequential sort of a thing, which is the way it is in eukaryotes. Here you can make buckets of it, like all in parallel. So it's a very good high output, very efficient system. But unfortunately, you don't have a lot of control over it. And that's the big name of the game in eukaryotes, right? Control. You like to have lots of control over things. So let's take a look at eukaryotic transcription. Starting off, first of all, with our polymerases. We don't have one, we have three. So Pol 1's job is to make ribosomal RNA, right? Or that's the structural component of RNA, of ribosomes. Pol 2, which honestly is the poster boy for most of our Pol studies, because they're the ones whose job it is to make proteins. They make mRNA and also snRNA, which is part of the spliceosome. We'll talk about that here in just a little bit as well. And then Pol 3 is going to be making tRNA and some other small RNAs, so some of those regulatory RNAs. But here's a good example. Here's a good question, right? So what creates the specificity? So how does a polymerase know which promoter to bind to? Because each one of these has its own promoter. There's an rRNA promoter, there's a tRNA promoter, there's an mRNA promoter. And the promoters are actually responsible for recognizing and recruiting the actual polymerase. So how do you know, how do you manage to keep, say, for instance, at Pol 3 from binding to an mRNA site? After all, it's all DNA, isn't it? It's all a series of A, C, Gs, and Ts. Well, that specificity is actually inside the promoter itself. So the promoter's job is to actually recruit the correct polymerase. However, it doesn't do this by itself. There's more. Of course, there's always more, right? So how does it do that? Well, in the initiation, notice the big initiation, right? That's the buzzword. In the initiation events, you've got an entire host, an entourage of proteins called transcription factors, whose job it is to essentially recruit, to first of all find the promoter. So that's the one thing they're going to do. They're going to find the promoter, and then they're going to recruit the correct polymerase to that promoter, and that's what's going to initiate the production of that particular gene. And then eventually, once they kind of kind of all come together, they form what's called the initiation complex at the promoter. So that's kind of like your initiation events. And then of course, termination, which is a little bit different, is there's no sequence. So there's not some sort of a stop sequence that says, okay, you're running this sequence, then stop. That's your termination signal. It's not like that in bacteria. Bacteria like that, but it's not like that in eukaryotes, right? So we don't really understand exactly quite as well how termination occurs. There's a lot going on in there. Actually, the Bentley lab over in Anschutz has done a lot of work on these events, these sort of terminal events of how you initiate termination, understand termination, and he's gotten a lot of work and a lot more understanding. We understand much more about what's going on here, but still, it's a lot messier. It's a little more stickier than just say like, oh, here, now stop, right? So it's not quite that easy. Of course, you notice there's one missing, isn't there? We go from initiation to what? And then what's missing? Elongation, right? And the reason why it's missing is because it's pretty much the exact same as we just saw in prokaryotes, right? There's only really one way to do dehydration synthesis, is once you get your initiation complex up and running, you still have a transcription bubble. You basically make RNA the same way by the same process, and you do exactly the same thing. So elongation is really not significantly different between eukaryotes and prokaryotes. However, initiation is, right? So here's an example of some of the transcription factors that are involved in this initiation, that is to say, recruiting the polymerase to the promoter. It all starts off with the first transcription factor, TF2D, which is this guy right here. That stands for transcription factor 2 because of Pol 2. That's because where we did most of our work. D, there's a whole host of them. I think we go all the way up to H in terms of the number of transcription factors. But TF2D has two major domains. It has a transcription factor binding domain, and then it has a TATA binding domain called TBP, or TATA binding protein. So the TBP, the TATA binding protein, is going to be located right in here. So you're going to have amino acids in there whose job it is to recognize and locate the TATA box. So it's the job of TBP to actually find and locate the TATA box, and there you land. Now then, your transcription binding domains are going to be located up here. And each one of those binding domains is going to basically allow one of these transcription factors to bind. And then what you're going to end up getting is this complex of transcription factors that have all been recruited to this promoter by TF2D. And then these guys will recruit the right polymerase. And then once this Pol 2 comes in and finds its group, right? It's kind of like showing up to a restaurant where you're meeting some friends there, right? You don't just walk up to a random table. Your group, your friends recruit you, right? It's like, hey, we're over here. So you recognize your crew and you go to that table because they're the ones you recognize. The same thing here. It's like these transcription factors are like the Pol 2 party. So they're like, hey, Pol 2, come on over here. We're over here at this table, right? So you kind of recruit Pol 2 specifically to this area. And then when Pol 2 binds in there, you have your final initiation complex. So these are events of initiation. Of course, after this, then you get elongation and then termination, and then we're done. But are we? Oh, no. It's just getting good now. We've just gotten started. So there's a couple of problems here, yes? First of all, after you do transcription, what you've got is basically a single strand of RNA. So we refer to as premature RNA or mRNA. Why is it premature? Well, think about it. There's some problems here, aren't there? You put this out in the cytoplasm, who's coming for you? It's like jumping into shark-infested waters. Right now, you're all nice and safe and cozy, inside the nucleus. As soon as you had the cytoplasm, who's coming for you? Your exonucleases, right? So that's a problem. Guess what? You have to modify your RNA now to protect yourself, because otherwise they're just going to gobble you up. That's the first step you do. That's the reason why you talk about mRNA modification. This is a eukaryotic thing. Prokaryotes don't do this, right? Because they don't have a need to do this. It's all right there. They don't have to go out there and scary, exonuclease land and potentially get chewed up. Okay. So the first thing you're going to do is create a 5' cap. And when you do that, what that does is basically does a couple of things. It protects that end, the 5' end, from degradation. So it keeps that Pac-Man from chomping it up. It also basically helps you in initiation. So this is oftentimes the binding spot. For ribosomes, where ribosomes have to grab onto this mRNA to get a hold of it, because that's what they're going to be reading. So this actually represents a binding spot for those ribosomes. Guess what? Now you have to protect your tail, your 3' tail, don't you? So that's where you're going to get into a poly A tail. So what you're going to do is you're just basically going to hang a bunch of A's on there. That has the effect of protecting your 3' side from the 3' exonucleases. So now you've protected your head and your tail. And so now you're in a much, much better shape, right? At least in terms of stability. There's also a lot of signals in this 3' end that are triggered by this 3' poly A tail that's associated with that termination events and things of that nature. So there's a lot going on in there, different types of processing and localization and things of that nature that's associated with that particular region. But the last big one is essentially the removal of non-coding sequences called introns. And so this is done by the spliceosome. So it's a process called preaminotic splicing. So here's the deal. What we need to do is we need to create what's called an open reading frame. What is that? An open reading frame is basically where you can start here translationally, start dropping in amino acids, and then continue through the entire sequence without interruption until you reach the termination. Okay, that's an open reading frame. That's not the way your pre-mRNA comes to you, unfortunately. The way your pre-mRNA comes to you is that you have typically these intervening sequences in between your coding sequences called introns. And then, of course, you have your coding sequences called exons. Okay. Now, in a lot of ways, what you get is a lot like having a sentence, right, of basically 3 letters, like the fat cat ate the big rat, right? Now, normally, how do we know where that sentence begins? That's subject, easier than that. The one thing that the English language has at the beginning of every sentence, it tells us it's the beginning of a sentence. Capitalization, right? The capital letter. That is your marker for this is the beginning. And then we start from there and we read without interruption through the sentence until we hit what? Some terminal punctuation, usually a period, right? So there's a beginning, there's an end, and there is a continuous reading frame where you can read one word after another without interruption, and it makes sense. That's an open reading frame. That's what we want, but that's not what we have. What we have coming off of the transcript is something more like taking a word, typing it out, and then smashing the keyboard randomly, and then typing the next word, and then smashing the keyboard, and then typing a word, and then smashing the keyboard, and then typing a word and smash. So different size smashes, but you have a bunch of random letters in between each word. Is that going to be an easy sentence to read? It's not. So what would you do in order to be able to read that? Well, you have to do more than that, right? Because you got a bunch of random letters in between the words.
Yeah.
So you'd have to go in there, find the random letters, right? Find out where the word begins, where the word ends. Find the random letters, delete those out and put the next word together, right? So you have to kind of go in there and delete out all the randomness, all the key pounding, and then sew it all together to create your open reading frame. Then you can read the sentence, right? That's exactly what's happening in this third one. We're going to talk about splicing. And it's driven by a complex called the spliceosome. We'll take a look at that one here in just a second. So here's your 5' cap, your 3' poly A tail, and then of course you have your code in between. That's basically a mature mRNA that's ready to roll. So that one now you can move out into the cytoplasm to essentially give it to a ribosome so it can start actually making protein. But what we want to do is we want to talk a lot about some of the splicing ideas, right? Because essentially you have introns, which are non-coding sequences. I usually depict those by a line. Exons, which are coded. These will be translated. That means to say that these will become amino acid, right? So that's actually code that becomes amino acid. These are usually depicted by a box. And generally speaking, when you actually have a gene, it looks something like this. There's exon 1, there's intron 1, there's exon 2, there's exon 3, exon 4, exon 5. So each of the intervening pieces are introns, the boxes are the exons. Now, generally speaking, when you snip out these introns, which do not become part of the protein, you use SNRPs to do that. And SNRPs are the actual protein RNA complexes that form the spliceosome. And their job, the job of the spliceosome, is to remove these introns. To do exactly what we just said, right? Delete out the random letters in between and then put the actual sensible words together. Okay, that's exactly what the spliceosome does. So essentially you have your DNA template. Here you can see these are your introns here. Here's your exons. So what's going to happen is you're going to take these introns, you're going to cut them out with the spliceosome, you're going to sew the introns together, or the exons together, and you're going to get, in this case, your open reading frame, your ORF. So you can start here, read all the way through, and then you're going to have your translated product. Okay. So this is kind of what it looks like. This is basically the same image. The only difference is we kind of give you electron micrograph that's showing you exactly what this looks like. So you can see how when you're starting to cut out these introns, you kind of form all these big loops and things like that. A lot of times students will look at this and be like, well, what am I supposed to make of all that? Well, this is only really important if you're really looking deeply at the mechanism of the spliceosome itself. This is just to kind of show you what it actually looks like, that it's actually things. So what happens is when you go to cut these introns out, you're really manipulating this mRNA, you're pulling it around and stuff like that. So you're creating all these big loops and things like that. So you're really physically manipulating these mRNA molecules. So that's kind of what it starts off with. The spliceosomal proteins, the SNRPs themselves, basically come in U1 to U6. There's about 6 of these designated U1 to U6. And all these proteins have the protein component, but then they also have the snRNA. That's where the snRNA comes in, right? Where that's a Pol-3 thing. And so you have several of them. A couple of them are basically responsible for locating the parts of an intron. Now, here's one of the things that I want to sort of make sure is clear. Because a lot of times students will be like, well, wait. Okay, so I see these boxes and these lines, and I understand that that's an exon and that one's an intron. I get that one. But how do you know where the exon stops and the intron starts? Because last time I checked all DNA sequences, it's a bunch of ACGs and Ts, right? Or it's, in this case, it's the RNA, it's ACGs and Us. Right, so the question is, how do you know? Right, how do you know where your exon intron boundaries are? Either on this end or on this end. And the answer is basically, we go back to consensus sequence, right? So there's actually, on the 5' end, what's called the 5' splice site of every RNA that has a consensus splice site. It has an invariable GT, and then the 3 after that usually vary. but that GT does not. Those first two positions are solid. Matter of fact, every intron and every gene in every organism across all taxa have the same GT consensus. So it is a hardlined GT. In addition to that, down here you have the 3' splice site, which is an invariable AG on this side with the upstream 3 being variable. So then right now, what you have is a pattern, right? You have an invariable GT. You go a little downstream, you see an AG there. That GT-AG pairing marks the boundaries of an intron. But there's more. Because down by the 3' side is going to be the branch point. This is a consensus A that happens to be hanging out there. So now you have 3 major recognition elements in the intron. If you see a GT and an AG with an adenine that's right just upstream of the AG, that is a pattern that screams intron. So the way that you recognize these sites is by these consensus sequences. That's great, but that still didn't answer my question. My question was, how do you recognize these sequences? Not what are they, right? Well, the way that you recognize them is snRNA that is inside the SNRP is complementary to the splice sites. So the reason why this U1 SNRP, which is what this guy is, can recognize the 5' splice site is because this snRNA right there is complementary to that 5' splice site. So U1 recognizes it literally by finding it in its snRNA and be like, ah, hey, there you are, GT, gotcha, right? And then there's other SNRPs that will come in here with identification, like secondary identification. It's like multi-factor identification, like with your bank or something like that. And then other SNRPs will locate the branch site. How do they do that? Do they just pick a random A? No. What they do is they complex with each other and they're feeling the AG. And if they basically feel that AG consensus, and then there's an A right about here, guess what? You've got yourself a 3' branch site. That's what you've got. Because normally what would happen is you would never have an AG here. You'd feel around, you'd be like, I don't feel anything. There's no A there. Guess what? You're not in an intron, right? You feel an AG, feel that A site, that branch site, boom, bingo, you got it. So that's basically what happens. You have a couple of SNRPs that bind to the branch site now as well. The next step after that is that these two guys will actually interact with each other. So these two groups of SNRPs will then interact with each other. When they do, they're going to pull all the mRNA with them. And that's where they're creating those loops. So here you can see they're all interacting with each other. You can see they're creating this loop. Then what's going to happen is they're going to sever that intron exon boundary there. They're going to cut this one here. And they're going to connect this one to the branch site. And that's the next step. So here you can see you now have this 5' end attached to the branch site. And you got this little lariat, which then goes off and gets degraded. But you now have loose ends, right? So now what you have to do is you take this end, and you just seal it together. And when you do that, you're going to create 2 exons that are sealed together. The excised intron will get degraded, and then your spliceosome can go back and do it again. Wow. That seems like a lot of work. Yeah. There's still quite a bit to go still too, because we haven't gotten to translation yet. Right. So this is that seems like a lot of work to get your open reading frame, doesn't it? Well, and that's kind of like the way we used to see introns, right? Introns, when we looked at them, we were a little puzzled because we're like, where do these things come from? Right? They all look the same. They all had GT-AG with the branch site. And we originally thought that they were ancient viral integration. Because one of the things that happens when you get a virus is it'll actually make a DNA copy of itself if it hasn't already. And then it'll actually integrate that DNA copy into your genome. And there it'll stay dormant, perhaps, for a long time, until one day it wakes up and then it reinfects you. Okay. This is actually something that we see a lot, like, for instance, in chickenpox, right? I mean, how many of you guys have had chickenpox? Not the vaccine for chickenpox, but actually had chickenpox, right? That was your vaccine for it, right? So for those of us who've had chickenpox, the virus got us, right? We itched, unfortunately. And what happened was that viral genome was integrated into our genome. So everybody who's had chickenpox has that viral genome integrated into them. And so it waits and waits until you get older, until it wakes up and it basically reinfects in the lysogenic cycle. And we know that condition better known as shingles, right? Because typically, as it turns out, the virus apparently likes to integrate into the genome of nerves. And so when it comes out, then it's attacking the nerves, which is the reason why shingles is kind of a nerve thing, more so than the original chickenpox, right? But that's exactly what's happening. And so the idea here is that introns came from this ancient viral attack because they all kind of look the same. And they all seem to be fairly random, because what they saw was that some genes don't have any introns at all. Some have buckets of them. I mean, 50 introns is a ton of introns. And you have everything in between. But it seems kind of random, right? But here's some problems with that. Now, that was kind of like the ancient evolution answer to the problem, which usually is always problematic when you're trying to talk about randomness, because random, as we noticed, the more you study biology, the less randomness is tolerated in biology, because things go catastrophically wrong when they become random. So there's a problem, a couple of problems with that one. First of all, is if you take a look at it, a viral integration into a genome is a big deal, right? Because you insert into a gene and you blow that gene up, and that could be a lethal event. Okay. We do mutagenesis screens doing that exact same thing. But then if you imagine that you have not just one integration system, but you have 50 in a genome, what do you think the likelihood of having a functional genome after being blasted by that many integration events? What do you think the likelihood of having a functional genome after that is? Nearly zero, right? Which is one of the reasons why the one argument I hate the most, that I think is the laziest thing that a lot of evolutionary biologists like to do, that just drives me up the wall, is they always like to say, but we're the end point. What you don't realize is that we're like the lightning in the bottle. And so we're that 1 in 600 million trillion quadrillion. It's kind of like the same stupid reasoning, like I am, therefore it's true. It's like, no, right? Because that's not actually a functional thought in my mind, right? Don't tell me about where the endpoint is. That's not what I care about, right? Tell me about how the endpoint came to be. And I can tell you right now, whenever we talk about randomness, it seems to fall apart every single time you study something that has an inordinate amount of randomness to it. It almost always falls apart because mathematically it just doesn't hold muster. And it requires you to actually put your science aside, which is not a good thing, ever, right? So that I have a problem with that one. I don't see a situation where that's a viable theory at all, because to have that many integration events would just do such catastrophic damage to the genome that it's nearly impossible for the genome to be functional after that. Not only that, but to compound that one, what we actually noticed was not only were these not random, but they seem to be intentional and organized. So we noticed was the exons actually are functional domains. So if the random integration of viral attacks is the theory, then you're trying to tell me not only did we manage to come away with a functional genome, but that all these random integrations happened by chance to actually separate the exons into functional domains. I'm sorry. I'm off. I'm not buying this bridge. Right? I'm not buying this bridge. So what this actually suggests is that there's a lot more, like the cell is like the master architect, and that there seems to be a mechanism that's driving a very intentional organizational process here, where you're not just using introns as some sort of a random grenade you're throwing at something, but you're actually intentionally placing these things in position with very intentional results coming out of it. Right, that's what that one says. Now, generally speaking, evolution, the idea is like, well, wait a minute, that was actually the organization came afterwards. Well, no, it didn't, because you didn't have anything functional after your integration storm to even organize it, right? Just because we rebuilt Dresden after the bombing of Dresden doesn't necessarily mean that was the intention of it, right? So there was nothing left of Dresden. We had to start over and had to be more of an intentional rebuild. And so, there's a question.
You don't have the same size.
No, the introns vary wildly, actually. They can be very short, they can be very long. So there's nothing about that. And that would also be another piece of evidence against the viral integration, because viral integration is largely uniformly the same size. Their genomes don't vary in size. They're mostly the same size. Yeah, it takes a copy of his DNA. What it literally does, it basically just kind of pops it right in the middle of your genome. Yeah, right. So if that was actually like dormant viruses, ancient dormant viruses that just aren't doing anything, and if they all started waking up, then we're talking about like a genetic storm of, I mean, unbelievable proportions. I mean, I would hate to go through whatever that looked like. That would be terrifying. And that's not even, there's other things actually that are in our genome, like mobile transposable DNA elements, which we also think are potential viral hangovers that have a similar sort of a problem to them. That are also scary, that have similar sort of an idea, is what happens all these things all of a sudden wake up. And we learned more about those in the genetics class, but they're really fascinating because it's like, oh my gosh, it's like the genome is like immensely complicated and fascinating, and it's like scary a little bit at the same time. Because you're kind of like, basically you're kind of cutting on like the fabric of life. What happens if you screw that cut up? Yeah, right. So it's kind of cool. Okay. So back in the day, the reason why I like to push back on this is because the way we used to view introns, and this is the one thing, this you imagine there's probably a visceral thing here going on for me, right? Because, and it's true, there is, because back in the 70s, they used to come up with a term, and they used to use the term junk DNA. And that's usually when they said that back in the 70s, they were referring to introns, which, I mean, I guess it makes sense, right? I mean, you cut them out and you throw them away, right? Therefore, it's junk DNA. The reason why I hate that term is because, generally speaking, that is a loser's bet. The second you say junk DNA, or the second you say, oh, it must not have a function, the first thing I think of, the first word in my mind is loser. You're going to be a loser. You know why? Because there's no such thing as a functionless anything in the cell. Everything is so costly that if you're not doing your job, you are not around. And that the other thing that drives me crazy is it suggests that just because we're too damn stupid to actually know what its function is, somehow its function is not important. Imagine, think about that for a second. Wrap your head around how immensely and profoundly arrogant that is. Oh, we don't really understand its function, therefore it must not really be that important. Yeah, that's because you're too stupid to actually understand its function. So you can park your arrogance somewhere else. By the way, guess what? We actually did figure out what the function was eventually, because there was enough people who actually didn't believe that kept on working on it anyway. This is one of the reasons why I already kind of told you, like one of the arguments that evolutionary biologists make that just drive me crazy. A second one, which is similar to it, also drives me crazy. And a lot of evolutionary people do it. And a lot of the old 19th century Darwinian people do it too, because all they did was study rock zoos and morphology. That's all they did. This idea of vestigial organs. Anybody ever heard of that one? Yeah, it's like evolutionary leftovers that don't have any function. Bullshit. There is no such thing. Because every single time we've considered that must be vestigial, must be an evolutionary hangover, we've later on actually pulled together our intellectual integrity, stopped being stupid and arrogant because we didn't understand the function of it, therefore it must not be a function. And we actually learned later on, whoa, wait a minute, this actually does have a function. We just didn't understand what it was. So when I hear an evolutionary biologist say this is a vestigial organ or it's a functionless holdover from evolutionary past, I think, lazy bastard, go do your job. Right? Stop doing the cop-out thing because you don't understand what it does. Go work hard and try to figure out what its function is, because I can guarantee you it will not be there if it did not have a function. Most vestigial organs, that's been true. But yeah, that was one of the original ones. That was one of my big pet peeves, because it is not functionless. It actually does have a function. It's part of your immune system. It's actually has it's related. It's related in spirit, not quite like the tonsils, but similar to. So it has a kind of an immune support. It's like a secondary immune organ. And the reason why we didn't know that is because, number one, evolutionary biologists were too lazy to figure out actually what it does. So we basically said, oh, well, this must be, a holdover from this or whatever. No, it wasn't a holdover. It's actually doing a job. It's just that we didn't know enough about immunology. But no, that was never an option back in those days. And see, this is my biggest beef. This is the reason why I say don't lose your objectivity. It's because it's like when you say something like that, you say, oh, well, this doesn't have a function. It's an evolutionary holdover. It's like, no, it's not. You just don't know what the function of it is. Tell me what the function of it is. And that function is no longer needed. And then maybe I'll buy that one. Right? But you just didn't know what the function of it was. It's not the same thing. So that's actually a false conclusion. That's actually a straight up lie. So when I look at that, I look at when evolutionary biologists, and there's a lot of them out there. Now I'm not saying there's no vestigial holdovers. What I'm saying is that the majority of what we consider vestigial hangovers are actually not. We just haven't understood their proper function and role just yet, because we're lacking the perspective, the angle, or the approach, or the tools in order to understand that function accurately. And so for you to say that basically it has no function is a lie, is a straight up lie. And that is what bothers me, because there's a lot of things to learn from it, but you need to be honest. And honestly, that's the biggest beat. That's the reason why I start off the semester by saying, don't lose your objectivity, because if you do, you become a liar, like a lot of evolutionary biologists out there are. Not because they're off course, but because they refuse to admit that I don't really know this. I'm just saying this because I don't want anybody to think that I don't know this. That's not good science, right? Theologians do that. Ooh. Now, if you're an evolutionary biologist, that one hurt right there. That was a kidney shot, and you're taking a knee on the tarmac, right? So, but I'm sorry, but that's blunt, but that's the way it is. Science got a lot of work to do, especially evolutionary biology. I started off saying I'm not afraid to poke the bear. I think what I just did gave the bear a right hook on that one. But let's take a look at these junk DNA proponents and actually ask ourselves, is it actually junk? Right? That's kind of where this alternative splicing comes into play. Is this junk DNA? No, because when you actually shut your mouth and do your experiments, it's just, I just don't stop, do I? Just like, I can't. This is
People need to hear it.
I love it.
this, I mean, it's, well, the reason why I'm kind of a little over the top is for an obvious reason. Because honestly, we live in a society that worships science. They worship science and they never question it. That is the worst thing on the face of the planet that you can do. You might as well join a cult if that's what you're going to do. Because science isn't about suppressing questioning. Science is about asking questions and always pushing back and always asking, are you sure? Are you sure that's how it works? Are you sure you know what you know? Because I'm not convinced. Show me. Don't just say it. Just because you write in a textbook doesn't mean it's true. Show it to me. Prove it to me. That's good science, right? The rest of it is just nonsense. So junk DNA, let's push back on it. So I say it's not junk. Prove it. Well, here's what happened actually. Around the late 80s, early 90s, right about when I came into UC Davis, there was a lab in California, a couple of them actually, from UC Berkeley, Jerry Rubin's lab. And from UC Davis, actually Ken Burtis's lab, they was actually right next to the door to the lab that I was in. I was in Scott Hawley's lab. And Ken Burtis was working on Drosophila, just like all of us were on that floor. But he was working on sex determination in Drosophila. So he wanted to know, what are the determinants that causes like maleness, right? So what causes the male fly to look like a male, like pigmentation deposition and all of its secondary sex characteristics. They kind of have this funny little thing on them, on their little forelegs called a sex comb. Males do. I guess that's, I don't know what they do with that. It's like a little friction thing to kind of grab onto the female when they're mating, right? But that's kind of like a little thing they got on their leg. And then of course, what are the determinants for female sexual determination? So the tissues, right? The actual tissues of the fly body. How do they become sexually determined? What they noticed was that there's two proteins. One was called double sex F, and so F for female. And this is a protein that if you were to express this in a fly, it would be enough to basically turn that fly into a female. Physically, the physical attributes of that fly would become a female. They also noticed that there's a double sex M, do the same thing. You express that in a fly, it'll turn that fly into a male. Even to the extent where if you've expressed that in a female fly, it'll convert that female fly into a male. That's how strong that determinant was. Very similar to testosterone and estrogen in us. But here's the thing. They knew this was double sex F and double sex M, right? So female versus the male determinant. But what they noticed was when they went to go find the gene, they noticed that they both came from the same gene. So that was actually the first evidence, those double sex gene. It's the same thing as the protein, right? So what they noticed was that the same gene was producing two different proteins. Now that's kind of crazy talk. So what I want to do is I want to go back to my little gene here that I drew. I kept it basic for a reason. Because when you're taking a look at alternative splicing, here's what alternative splicing actually is. So at every 5' splice, so you have a GT, you have an AG there. You have a GT there, you have an AG there. You have a GT there, you have an AG there. GT and AG. Now, generally speaking, what will happen is you basically cut out, you pair that GT with that AG, that GT with that AG, that GT with that AG, that GT with that AG. What you produce is after you seal everything together, an X1, X2, a long exon 3, a long exon 4, and an exon 5. That's your open reading frame. But a lot of times students will ask me a good question. They'll say, well, wait. So if you're pairing a GT with an AG, is there any reason why you have to pair that GT with the immediate downstream AG? Could you possibly pair up this GT with this AG? And the answer is you could absolutely do that. So what happens if you do that? That means you're going to basically sew together exon 1 with exon 5. Different open reading frame, right? What happens if you pair up this GT with, let's go, this AG? So now you're going to do this cut right there. So that's going to give you an exon 1. 2 and 3 are out. Whoops. There you go. You're going to have a 4 and a 5. 3 different open reading frames from one transcript. producing how many proteins? Three different proteins. Isoforms, yes. So an isoform is basically a protein or proteins that are related to each other by the fact that they come from the same transcript. They've only been alternatively spliced. So notice you have different patterns of splicing. Why is that a big deal? Because not only did it seem like the introns were intentionally mechanistically placed to separate the exons into domains, but now you can skip these exons and you can create multiple versions of this protein. So different proteins from the same transcript.
Let's say on that, when you put together X1 and X5, what happens with 2, 3, and 4?
They're gone. They get part of so everything in between is part of the lariat, and that gets degraded. So you can see that if you're going to call introns junk DNA, then during alternative splicing, you got to call all those intervening exons also junk. And they're not indeed, nor were the introns. Why? Because you wouldn't be able to produce these three different proteins if you didn't have the exons there to give you these patterns. So what we found out was that in the case of double sex, and this was kind of the first, I remember actually, this was still, because I got to UC Davis around 1996, and this was even at that point still a very, very heated debate. But this was the pushback. The idea was like, listen, the reason why you have two different forms, DSXF and DSXM, is because they are alternative products of the same transcript. Double sex makes one transcript. In females, the splicing pattern gives you double sex F. In males, the splicing pattern, alternatively spliced, gives you a different mix of exons, but it gives you DSXM. So sex, and the reason why this hit home was because the idea was, this is not consequential. But this proves that, first of all, it's a thing. Second of all, it's important. Third of all, it's not junk. Fourth of all, it's a significant mechanism. We're talking about sexual dimorphism in fruit flies. Whether or not you are a physical female or a physical male is all founded upon alternative splicing. After this experiment, the term junk DNA disappeared quickly because no fool on the face of the planet would mention that again, because it would make them look really stupid. And of course, researchers are too insecure to be thought of as wrong. There's a reason why they would rather lie than to admit they're wrong, because there's a lot of ego in research as well. Unfortunately, that's not a good mix. So when we take a look at this and when we kind of fast forward then to the Human Genome Project, roughly not quite 10 years later, around 2000, we were expecting, and this is a big question the genomics people were asking, right? They're asking, well, how many active genes do we have to make this, right? The whole human phenotype. It's got to be hundreds of thousands, right? It's got to be, think about how many proteins are in the body. I mean, there's like tons of them. You're talking about one gene per protein. You got to be talking about hundreds of thousands of genes that are firing at the same time. But when we actually came out with it, the number was between 20 and 30,000. Now, if you're a genomics person, and this thing I love about genomics is because when we came out with some of this stuff, I mean, our collective jaws hit the floor. And we're still kind of in the period of we're trying to clean our jaws up, our teeth up off the floor. I mean, we're just still completely, I mean, just mystified in a lot of ways, because there's some amazing stuff that's come out of genomics that we're like, don't have an answer for this, right? And this is one of them. It's like, wow, not only were we off, but we were off by like orders of magnitude. So how is that possible? It seems like it's wrong. This is one of those things, by the way, when science gets cute and they say it's like, oh, obviously it's wrong, right? So when you did all of this experimentation, it was obviously wrong, because that can't possibly be right, because why? because that would mean I'm wrong. And I'm too insecure and emotionally frail to admit that I'm wrong, instead of scientifically robust and objective to admit that my hypothesis may need modification. Okay, I'll stop patronizing. See, now I've gone from poking the bear to like teasing the bear, right? So you see, I'm not afraid of the bear, basically. It's whatever, right? But this is hugely underestimated, right? Because now all of a sudden you're thinking, whoa, wait a minute. We're off by nearly orders of magnitude, right? So we basically take a look at 20 to 30 genes, and it varies a little bit. And you're talking about those are spitting out, 100,000 proteins. Why? Because of alternative splicing. What we noticed was that a lot of genetic disorders we were discovering were actually alternative splicing disorders or splicing mutants of one strap or another. And none of that, but we actually take a look at the human genome and say how many genes exhibit alternative splicing. We're looking at between 1/3 to 2/3 of the human genome is exhibiting some form of alternative splicing. What that tells me is that alternative splicing is the rule, not the exception, right? Which makes total sense. I mean, isn't that like in complete harmony with the way the cell works, right? You try to take something and you try to double your productivity, triple your productivity, quadruple your productivity. I mean, the cell's always doing that. And we learned that throughout the semester. So then we picked our jaws up off the ground. So now once we do that and we kind of create our open reading frame, now we're ready for translation. So let's go ahead and take a look at translation. First of all, we need to be introduced to some of the workers associated with translation, obviously the ribosome and the tRNA. We're going to start with the tRNA, then we're going to roll to the ribosome. So the tRNA is kind of a cruciform structure molecule, which on one side is going to have what's called an acceptor site or an acceptor stem, whose job it is to bind to an amino acid. Then on the other side is going to be what's called an anticodon loop, which is going to have 3 nucleotides that are going to be complementary to mRNA. So the mRNA codons are going to have a complement to them in the way of the tRNA. So the anticodon is the complement of the mRNA codon. Okay. So now you have both of these particular situations, and oftentimes the question comes up, where does the assignment, the specificity for the amino acid and the anticodon come into play? So basically, so where does the specificity come from? So where does the specificity of the assignment of the correct amino acid to the correct anticodon, where does that come from? So for instance, when we looked at Nuremberg's work, right, he noticed that UUU as a codon, mRNA codon, basically was phenylalanine, right? Who gets to make sure that selection is made? It's actually this enzyme right here. Aminoacyl tRNA synthetase is the one whose job it is to make the correct assignment between the amino acid and the actual anticodon itself. So this is kind of some representations of tRNA. So you'll see you've got the clover leaf view of things. And I like this one because you can see it's single-stranded mRNA or RNA, tRNA, that's folded up on itself. And it forms these stems and loop structures. Now, these guys are important because it's with these that you bind to the ribosome itself. So that's actually what's going to be binding to the ribosome. And so your acceptor site is up here. And of course, your anticodon loop is down here. You have your ribbon molecule, your space fill, and then of course, this is the one we're going to be using, the cartoon version. That's basically what we're going to be using mostly for the rest of the chapter. Okay. So let's go ahead and take a look at the charging reaction. So when we take a look at the charging reaction, the first thing you need to do is basically figure out how to take the correct amino acid and attach it to the acceptor site, which is essentially going to be utilizing ATP. Remember, nothing is for free. Everything requires energy. And you're going to be bonding this amino acid to the tRNA at the acceptor site. So that's basically what you're going to have. So this is the mechanism. So here's our aminoacyl tRNA synthetase, this guy right here. It's got 2 active sites. The first one is for the amino acid and we pick tryptophan as an example in this case, okay? And then the second side is going to be for the tRNA. So it's got two active sites. Now what happens first is hydrolysis of ATP. So you can see ATP is going to hydrolyze. It's going to leave behind AMP, adenosine monophosphate, right? Because you lose your two phosphates, and that's going to leave as pyrophosphate. And then what this does then is it tethers the tryptophan to its active site. So that's what the AMP does. So now that it's bound, then you have your tRNA, which will be able to come into and bind to its active site. But here's a question. Notice this particular tRNA is for tryptophan. But how exactly do you get the right tRNA? Because all the tRNAs look the same, with the exception of their anticodon. So the question is this. How do you ensure that the correct tRNA is bound to the site? How do you know that the phenylalanine tRNA doesn't sneak in there and sit down in there? That would be a misassignment, wouldn't it? Well, the way that this actually works, and this is kind of the big version of it. So there's kind of this one here. So the way that this has worked, the correct tRNA is selected by what's called an induced fit mechanism. Now, generally speaking, in enzymology, what we oftentimes notice was in enzymology, we kind of sometimes look at it as either a lock and a key type of thing, right? So it's kind of like these chairs here, right? They have a predetermined shape, and that shape is designed to fit your body, right? So that would be like a lock and a key. It's like a predetermined shape, and your body just sort of fits in there like a square peg, square hole, right? And that's the way we used to think of enzymes and substrates, but that's not actually the way it works. In reality, what induced fit basically says, it's a beanbag chair mechanism, right? Okay, that means explaining, I think. So does a beanbag chair have a predetermined shape? No, it's an amorphous blob, right? But as you sit in it, all of a sudden, the shape of the beanbag chair changes, right? And it becomes synonymous with the shape of your body. So the beanbag chair takes shape, but only as you are imposing your shape on it. So the shape of the beanbag chair is induced by you. So you sit on it and you're inducing a three-dimensional change in the beanbag chair that fits your shape. That's induced fit, okay? It's like sticking your hand into a marshmallow, right? So it'll basically adapt whatever shape you put into it. The same thing is true here. In this case, the induced fit is associated with the tryptophan binding. So with the tryptophan binding causes a shape change. And the shape change changes the tRNA shape so that, which makes the tRNA site specific. Think about it. It's the binding of the amino acid that induces a shape change in the aminoacyl tRNA synthetase. That changes the tRNA site to only allow the correct tryptophan tRNA to bind. So when you ask, whose job is it to make sure the assignment is correct? It's actually the amino acid is the one who's responsible for selecting the correct tRNA. Because the amino acid is the one that changes this site to only correlate with the tryptophan anticodon. Same way, phenylalanine will come in, change the site, but the site will change differently. Just like somebody else who sits in the beanbag chair will create a different shape in the beanbag chair because our bodies are all differently shaped. Adelia.
say the codon got changed instead of amino acids. Got changed. How would that? That's the right amino acid.
Yeah, so that'd be a problem. Right, so as it turns out, you can see that there is a little bit of, not all three are represented here, right? So we'll talk a little bit about something called wobble pairing here in just a second. But there's kind of a little bit of slop in this, a little bit of flexibility, especially in that third one that will accommodate that. The other thing is also when you mutate it, what did you mutate it to? Because remember, everything about the tRNA is the same. So if you mutate it to a different codon for a different amino acid, guess what? You're basically shaped like that amino acid. So you're going to pick up the wrong amino acid because now you've basically become a different type of thing. Okay. Generally speaking, like when it comes to mutations and anticodons, we don't see a lot of that actually. Because it's like either you fit in there or you don't. And if it's very, very exclusive, then it's like you just don't get charged. And so it's like an absent. So you kind of absent yourself out. But the problem is there's other tRNAs. So you're not the only one. So there's other tRNAs and they're probably covering for you. So in order to see like a global breakdown, say tryptophan, you would have to basically mutate every single tryptophan tRNA gene out there. And typically you have multiple copies of these. So that's the reason why if this one doesn't work, so, you know, then the ones that do work will basically take over, right? So you either won't fit, or if you do, it'll be a misdelivery. You could get a misdelivery. But in that case, you'll see that come out in the protein probably as like just a bad amino acid, like a different amino acid. It's like, whoops, that's a wrong amino acid there. So it kind of depends. It's a very situational kind of a situation. You have to really look at the specifics of it. Mutations, even though we talk about them generally, can be actually very specific in nature and the way they behave. So then, of course, at this particular point, then what happens is AMP is going to leave, and that's going to be the stimulus for actually connecting your tryptophan to the acceptor site. And at that point, you're ready to roll. So you get released, and then you're off to go to translation. Okay, so let's take a look. That's tRNA. Now let's take a look at the ribosome. So the ribosome, when you take a look at it, actually has three major active sites in it that are important. The first one is what's called the P site. This is the one that's going to have the growing polypeptide chain, and we're going to take a look at this actually. When we look at the mechanism of translation, it should make a little bit more sense than it does right now. So that's one major site that we're going to see. The other major one is called the A site. I refer to this as sort of the arrival site. So this is where the new codon that's supposed to be read for the next amino acid in line arrives. So it's kind of an empty chamber where that new amino acid will arrive. And then the third one is the E site, the exit site, or the ejection site. So this is where the empty tRNA that just got spent is now uncharged. And so it gets kicked out and has to go get recharged. Where does it go? The aminoacyltRNA synthetase to go pick up another amino acid, right? So it goes right back to the same mechanism we just saw to get itself recharged. Okay, so here's kind of what it looks like. You can see you have the large and a small subunit. The large subunit is going to create this EPA site. So where you have your P site in the middle. Notice in each chamber, you've got one codon existing in each chamber. Right, so here's your exit site, your polypeptide site, and your arrival site, and your mRNA basically kind of threads through the bottom of those chambers. Okay, which is exactly the size of a codon, and this is basically the structural look of the ribosome. So the one thing about the ribosome that I always like to stress is the ribosome does one thing and one thing only. It basically forms peptide bonds. That's all it does. It's actually a process called peptide transfer, and it's driven by peptidyl transferase. So all it does is basically create peptide bonds. That's all it does. Doesn't do anything else. And it'll do that with whatever you give it. So if you give it the wrong amino acid, it doesn't check it down. There's no quality control associated with the ribosome itself. All of that is done with the aminoacyl tRNA synthetase. Okay, that's the one that makes sure that those assignments are correct. So if you want to give the ribosome a bad amino acid, it's just going to take that one and add it to the list. It just add the growing chain. Okay. So let's take a look at translation in prokaryotes, right? Because we want to start with prokaryotes and we'll basically transfer that over to eukaryotes. So when you take a look, the first steps are going to be with initiation, right? So what's the initiation of prokaryotic transcription or translation rather? So the first thing you need is your first tRNA, which is always the same. It's N-N-formal methionine. You also need the small subunit, and you need your mRNA with your start code on it, right? So that's the idea. You have your start code on there. So in order to assemble all this, these are the three pieces of your initiation complex that you need. In order to assemble all this, you need a place to actually tether your mRNA to the small subunit. And bacteria do that with a sequence called the ribosome binding sequence. So there's actually a piece of sequence in there that sort of is a binding signal to say, here, bind here. And that'll allow the small subunit to bind there. And then once that's bound, then you basically cap it with the large subunit, and that'll create your EPA sites. So that's kind of what it looks like. So here's your small subunit. So you have an RBS right here. And that will basically bind to your small subunit. A couple things I want to point out. This is your initial tRNA, the first one, right? That's your N-formal methionine. But remember, these things aren't just coming together randomly. They're not just floating together. They're not just randomly sort of bumping into each other, right? Remember, nothing is random. These guys are actually being escorted, and they're being very escorted very intentionally. So notice here, you have an initiation factor, a protein, that's basically assembling that mRNA into the small subunit. You also have another initiation factor here that's actually shuttling your first tRNA into place. Think about it. Let's go back to performing arts. One of the things you'll notice in performing arts, sometimes if you see the stage from above, is oftentimes you'll see taped like marks on the stage. Sometimes X's, depending on if it's a lot of marks, it's complicated. Each person will have their own symbol. And so the idea is when you start off a scene, you hit your mark, right? So you stand on your mark. This is where you're starting. So you want to make sure that you're starting off on the right foot and you're in position for the scene to take off. But typically, when you're looking, if you look carefully, what you'll notice is that invisibly behind this, making sure all the major characters and actors are in the right place at the right time, you have invisible stagehands, right? Because these are the people who are quietly behind the scenes, making sure that each of the actors knows their mark and where they need to be at a particular opening of the scene, right? And so you never see it. If it all goes well, you never see the stagehands, right? Because their job is to assist the actors, making sure that everybody is where they need to be, when they need to be there, so they can actually do the scene. The exact same thing is true for these initiation factors. So these are usually invisible. You don't really see them, right? But they're there making sure that everybody is where they need to be. The mRNA is where they need to be, binding to the small subunit, that the initial tRNA is where it needs to be, coming in and binding to the start codon. Once it all assembles together, you'll notice you have your mRNA now thoroughly bound to your small subunit, and you've got your initial tRNA attached to your start codon. Guess what? You're ready to roll now. Then all you do is cap it with your large subunit, and now you can see everybody is in position, right? Your P site is carrying the polypeptide chain at this point, one. It's got one amino acid in there. Your arrival site is empty, which it should be, because this is the next codon in line that needs to be read. Your exit site, in this case, is empty. Why? Because this guy gets kicked out so he can go get recharged and bring in another amino acid. So now once you get to this point, this is your initiation complex. After this, it's time to elongate. So that's in bacteria. What does it look like in eukaryotes? Well, it's very similar, actually, with the exception of we don't have N-formal methionine. We just have regular methionine. And instead of having RBS, we don't have an RBS. What we do is we use our 5' cap. Remember, bacteria don't do the 5' cap thing? We do. So the 5' cap actually helps us to bind to the small subunit. So similar function, slightly different thing, right? So that's a convergence of function, but a divergence of structure. So once we actually initiate all this, we have our ribosome capped. Everybody's in position. Now we're ready to go. It's time for elongation. Important thing about elongation is basically you're going to start now putting together and creating the chain of amino acids, our polypeptide chain. You're going to do that with, in this case, elongation factors. EF2, elongation factor, TU2, is a big elongation factor that will help to actually bind to shuttles tRNA into the A site. Notice we're not just randomly drifting. We've got an entourage. We've got a shuttle. Somebody's making sure that we are on our mark at that particular time. Then as our new tRNA arrives in the A site, we basically transfer that amino acid to our growing chain. Okay. So what I want to do is I want to end with this and at the same time start with this, because this is our 3-step mechanism to elongation, which starts with codon recognition, peptide transfer, and then translocation. And so I just want to sketch it out, right? These are the 3 steps of elongation. And so we're going to start off and we're going to talk a little bit about codon recognition, what we do after that, and then what we do after that. So we're going to go through each one of these steps, take a close look at what's going on in each of them. And then once you finish the cycle, the nice thing about this is you just do it all over again, right? And so this is a good place to start. And then probably we'll easily finish the chapter on Wednesday.
And the test is Monday?
Yes, the test is going to be Monday the 8th. So the test is going to be Monday the 8th. Yeah, that pesty test is going to be Monday the 8th.